Okay, as you can probably see, I'll usually start most lectures off with a new slide that says questions. So does anyone have any questions? The notes?
Yeah, the go over
Yeah, we're just going over organelles today, so you should already have that. Yeah, and we're going to talk about the first exam a little bit. No questions?
Is water not going to be on?
We'll see how far we get. We're about 2 lectures behind, and this is why I put this up. So this is from the syllabus. We're about 2 lectures behind, so we'll see where we get to. We might get through just the next part, which is basically molecules. And so I will have you some handouts, and the handouts will be more extensive than what we cover. And one thing I keep on to emphasize, if we don't cover it in class, then don't worry about it, okay? It's just this book does not do a really good job of covering protein structure. So I'm going to give you some handouts. It's just chapters from other books. They'll be more extensive, so you don't have to worry about the stuff we don't cover. And then after molecules, then we go into water relations, and then we move up from there. Please, if you want the survey as of now, oh goodness, 50% for Wednesday, I think. I may have the day wrong, but there was one day that had more than the others. But I'm probably going to do like a set session on one day, and then just have open office hours, and also you can use the Hangouts to talk to me. I'll go over that more. There's instructions, of course, for you if you're online, or for you guys in here to set up your accounts and get the Hangouts working. I'm not going to spend a lot of time going over that until we start to do it, because no one, based on my experience, no one's going to do it until they need to do it. So I find, especially with software, if you try to use it and then someone shows you how to use it, you go, oh, okay, that makes sense. But if I show you how to use it just from square one, you don't remember. I don't remember a thing. Someone will say, now push this, now push that. You go, now what do I do? Oh, I don't know. So it's not that big of a deal, and they'll be broadcast and also be recorded. Okay. Okay. So today we're going to finish up probably about halfway through the time, maybe not. And we're talking about organelles, and I wanted to pick back up a little bit before we finished. And we've talked about the vacuole, we've talked about most of the organelles, and I wanted to go back over this picture, this micrograph, and sort of give you a feel again for what a mature, in this case, leaf cell looks like. So it's mostly water. And it's the water that's pushing into, and you're going to get this one. I tell you everything several times. When we get into minerals and we get into water, you'll understand this more. But basically, you have water coming into the cell, and this is the vacuole, of course, and then that causes water or pressure to press against the vacuolar membrane, which then presses against the cytoplasm, which presses against the plasma membrane and presses it against the wall, okay? So for a plant to be fully functional, there is water, there's pressure pushing against the balloons. Okay, the balloon inside the balloon. And if we take that cell wall away, what happens? Boom. We have an explosion. Because water will expand the membrane to the point of where it's going to explode. It's just going to break. Okay? And you can put things in and out through, I've even seen somebody at a seminar where they talk about how strong, they actually blew a balloon up and put a pin into the balloon without causing the balloon to rupture. And the point was you can have these really small holes, you know, the plasmodesmata, where you can move stuff around from cell to cell without crossing a cell wall, number one, and without crossing a cell membrane. Something you need to start getting is that most things move into the cells through protein transport. Even water has transporters. Water can diffuse through the cell wall, of course, and through the cell membrane. But most everything that moves through cells are carried by carrier proteins. Okay, and you'll get that again and again and again. Okay, we're going to talk about chloroplasts and mitochondria today. There is an assigned reading, and I'll send that out to you from the book, from the Hopkins book, about the kinds of chloroplasts that, or the kinds of plastids that are found in different kinds of cells. Someone sent me a question, and right now I'm still a little bit out of it. So if you send me a message, it might take me a while to get back to you because it's not ringing on all my computers, so it depends on where I am. So we talked about we had the nucleus, which was a double membrane bound organelle. It has two membranes around it, and the outer membrane basically becomes the endoplasmic reticulum. Okay? Now we're going to talk about two organelles today, the other two organelles that have two membranes around them. And that's the chloroplast and the mitochondria. As I mentioned before, and I will mention now, these organelles are strange, I guess they're not strange. They have their own DNA. And they actually have bacterial ribosomes. So if you squint your eyes and look at it, it looks like a bacteria. And you don't need to know the kind of ribosomes, but you know there are different. We have what are called 80S subunit ribosomes. Bacteria have 70S, and guess what? The chloroplasts and the mitochondria both have these bacterial kinds of ribosomes. So we would hypothesize that maybe some time in the glacial past, these were bacteria that slowly became incorporated into what then became green plants. And as an aside, when you see people doing a lot of analysis of especially older DNA, they're actually looking at mitochondrial DNA. And you can actually go back and you can recover mitochondrial DNA from very old samples. And so when you see this work done where they're tracing people back for thousands of years or even hundreds of thousands of years, it's all mitochondrial DNA. And the neat thing about mitochondrial DNA is that comes from your mom only in plants and animals. So you can track things back genealogically, okay? So we all know what mitochondria do. We all know what chloroplasts do. You're going to know more than you want to know about what mitochondria and chloroplasts do. And I think I want to go over this again because I want to add a little bit of what actually goes on in these organelles. And you have this inner and outer membrane, and the outer membrane is fairly leaky. In other words, things can sort of go through the outer membrane. And then the inner membrane has all these folds. Now, what's the answer to why there's all these membrane folds? What's the purpose of that? Surface area. You get a lot more surface area in a small amount of space. And if you know what electron transport is, there's both photosynthetic and mitochondrial electron transport, very complex. It happens on this inner membrane space. You'll get electron transport in a while. So we have this outer membrane, and then we have this folded inner membrane, and these folds are called cristae. And then, I think I told you last time, the big deal about mitochondria is they have these motors called ATPases, and this is where almost all of the ATP is made for the cell, both plant and animal. Okay? And they're motors. And I'm actually going to show you a picture of the motor actually turning around later on when we get into respiration. This is where oxygen is used. This is why 99.9% of the reason why plants and humans, animals need oxygen is because of the mitochondria. One single step that you're going to see. One, you may have heard it called the electron acceptor or the terminal oxidase. Don't worry about that now. And guess what looks like oxygen? What molecule would you not want to be that looks like oxygen? CO2. Not CO2. CO does, but even worse is a molecule we call cyanide. Cyanide looks like oxygen. When I say it looks like it, the enzyme can't tell the difference between oxygen and cyanide. So at any given time, you've got a 5-second supply of ATP in your body. If I disrupt ATP production, you die. Plants, on the other hand, they're used to being flooded, so they can survive flooding a little bit. Okay? But most of the reason you need it is because this is where most of the ATP is used. This is where the Krebs cycle is, or the TCA cycle. What else is it called? Krebs cycle, TCA. There's another name. Anyway, you probably heard mostly the Krebs cycle. That's where those enzymes are found. A question I often ask is, I give you a container that contains mitochondria or chloroplast. Without looking inside, tell me how you could tell the difference between the two. Okay? And as you go more into the semester, you'll have more and more reasons. We'll keep coming back to these. Okay? Questions about this? So they have DNA, ribosomes, they have protein synthesis. There's sort of these worm-like things. You get this idea from these pictures that there are these nice little things. Remember, everything in there is moving around. Everything's not static. So you got these things moving. They're not just like, they're membranes. And membranes are liquid. In fact, next week I'm going to talk about what happens when membranes are not liquid, which is what's going on last week and part of this week. If a membrane becomes solid, it's a dead cell. Okay, so think of the difference between lard and oil. What's the difference at room temperature between lard and oil? Lard's solid, oil's not. If your membrane is more lard-like, you're dead. So everything in here is just moving around, you know. You've probably seen Brownian movement. Remember from either high school where you would take the leaf cell and put it under a microscope and you'd watch the chloroplast move around? So everything is liquid. The membranes are liquid. The only thing that's not liquid is the cell wall. Questions? What is this thing? That's chloroplast. There's the parts of the chloroplast? What are these big old dark things? Anybody know? You find them in chloroplast. Huh? Those big old dark things are starch. Well, the grana is the little things I was pointing to. These little stacks are the grana. We're going to see that just here in a second. What is this right here? That's a Golgi. You know, I think I told you I had a course where on Friday we were given a micrograph, and the person that could identify the most organelles got the highest grade on Monday. So it was real. So I learned how to look at a lot of micrographs. It's fun. Yeah, I know you think it's fun. Okay. Now let's go to the other bacterial type thing we find, and only in plant cells, and this is a plastid. In the reading I'm going to have you guys do from the book, it's going to talk about the ontogeny of the different kinds of plastids. There are more than just chloroplast in plants. There are specialized other kinds of plastids. Now we're talking about an even more complex inner membrane. So in the chloroplast, you have the outer membrane, and then you have inside this inner membrane. And guess what? It also has an ATPase. So there is the notion, of course, that way in the distant past, this bacteria sort of became something with maybe an ATPase, and then in a tremendous amount of time later, maybe something like a mitochondria, and then something like a chloroplast, and they started then over, glacial time, differentiating into what we know as mitochondria now and chloroplast. Makes sense. They produce energy in just about the same way. One uses glucose, the other one uses sunlight, right? And we'll go into detail about those. Now, they're this very, very, very complex structures in this inner membrane. And there's an inside and an outside, okay? First, let's talk about what this stack is called, grana. So this stack is a grana, okay? And then collectively, all the inner membrane is called thylakoids. Now, in this cross section, they have blown up a single one of these stacks. And what you have is an inside and an outside. So see the yellow? This is the inside. That's called the thylakoid lumen, or just the lumen. Okay? And something I forgot about the mitochondria I need to back up and mention. Yes. The lumen is specifically for the chloroplast. And in this cartoon, you have a lot of protons in here. And what you're going to find is that protons are used to make ATP. And then the soup that's inside here is called the stroma. Okay? So the light harvesting, photosynthesis, the actual energy absorption occurs in these grana. Now, why are they stacked like this? Two reasons. I'll give you one reason. It's increased surface area. But why are they stacked like this? Absorb light. They're not green. They're clear with little specks of chlorophyll in it. Now, we see a tree being green, and I told you last time that no, it's not the tree that's green, it's the chloroplast. Now I'm telling you it's not the chloroplast that's green, it's the little specks on the grana that are green. The actual chlorophyll molecules, so there's a lot of them, but the point is that the light hits here and it can go on down in here before it's absorbed. So it's not just the light will be absorbed all through these stacks. So if you could look at it on a very, molecular scale, you'd see a clear membrane with the chlorophylls and the carotenoids and all the other. You'd see individual specks, okay? So that really is the only place that a plant's green. And then the stroma is where some of the reactions occur. And I want to back up here because I forgot one thing about the mitochondria, I think was the matrix. And the matrix is the soup inside the inner membrane. And that's where the actual TCA cycle occurs. It's in the soup. I'm sorry?
What cycle?
TCA, Krebs cycle. They change the name every few years. It was the Krebs cycle because that's the guy that found it. Then they changed it to, it's different things in different books. I really don't know what your book calls it. I just say Krebs because almost everyone knows what that is. But TCA, and then there's another name for it. I can't remember. So the matrix, and I forgot about that a while ago. Now we're looking at a chloroplast. And all of this now, let's go back here. The stroma, the thylakoids. So these are all connected. And then these really dark areas here are the actual grana. And then you'll also find sometimes these little inclusions in here. There could be different kinds of inclusions in there. Don't worry about those. And if you look at this, you can see that this would be the vacuole, and you see this little tiny layer of cytoplasm around it. So here you have the cell wall, a little bit of cytoplasm, and then the chloroplast. And then these darker areas are the actual place where the light is absorbed, okay? So the plants are green because there are little specks of chloroplast in those grana membranes. Now there's a lot of chlorophyll in these grana membranes, okay? That's the only place that's green. So most of the chloroplast, the outer membrane, is basically clear. Questions? Again, two membranes, the outer membrane, the inner membrane. In both the mitochondria and the chloroplast, you have this amazing amount of differentiation of the inner membrane. For the same thing, producing energy, for the most part producing ATP, using two totally different mechanisms. And the one, of course, unique to plants is the chloroplast. Now, this is the reading. It's a box 5.1 in chapter 5. And one thing you may not know is that there are different kinds of plastids in plants. And mitochondria and chloroplast, mitochondria and plastids come from other mitochondria and plastids. They actually divide. That's where they come from. So if we look at a parenchyma cell, which is, remember, an undifferentiated ground cell, wherever in the stem or whatever, it may have something called a proplastid in it. And this proplastid, depending upon where it is, can develop into different things. If it's the red part of the tomato fruit, it can develop into something called a chromoplast. depending on where it is. So the red in a tomato fruit is due to red color inside this kind of specialized plastid. Now, if you look at it, the inner membranes are sort of there, but not really. It's just basically a little, it's just a plastid filled with color. Okay? If we, no, chromoplasts are not photosynthetic. In fact, one of the most common things you can do with tomatoes, if you pick them, if you pick them commercially, is they can actually get sunburn. Think about where tomatoes are produced. They're produced down under the leaves. So you have to be careful commercially. If you in the middle of the summer in Texas, you pick them, they will absorb, just like we do, they'll absorb excess heat energy and be damaged. Light energy and be damaged. Then if we go to the other end of the plant, if we go to a potato, not a plant, the actual storage organ, then it's filled full of plastids called amyoplast, and the only thing they do is store starch. Okay? And there can be kinds of plastids in between these, but there are multiple forms. They all come from these proplastids, and like mitochondria, they come from other plastids. Okay? Now, both mitochondria and the chloroplast needs the nucleus. They're not standalone. They still need stuff from the nucleus to operate. But for the most part, they do replicate. In some cases, in fact, in the case of chloroplast, light is required. And there's something called a teleoplast or an idioplast. Anybody know where you find those? If you grow seedlings in the dark, they grow pretty high. They're trying to get to the sun. And what they have are plastids ready to go. All they need is light, and they quickly become chloroplasts. And you can actually, this isn't a great picture, but you can see they have sort of like this crystalline-like structure in the center. Give it a little bit of light, and boom, it turns green. So if you've ever done this, one of the hardest things we can do is grow plants in the dark and work with them, because just a little bit of light will cause them to green, right? I mean, you've probably all seen that. Or you have potatoes under your counter, you left them there too long, and you pull them out, and you've got the white sprouts growing out from them, and a couple hours later, they're already starting to turn green. So they're ready, the chloroplasts are there ready to go. It's a survival mechanism. They don't wait for the green light to start going from proplastid to chloroplast. They go to this pre-chloroplast stage, so that only a little light goes boom, and they're green, and they're now producing energy. Okay, questions? Okay, well, let's finish up here with a couple of pictures. What is this thing? Not a vacuole. Good guess, though. That's a nucleus. This is what will be the vacuole, these little things here. This is a meristematic cell, very densely packed. There's some mitochondria. You can see the nucleolus in here. And here you see the cell walls, primary cell walls. What are these things on this side? Worm-like things. That's a mitochondria. This is what probably will be part of the vacuole. Mitochondria. What is this thing and this thing? Golgis. Yep. Here's a little bit of endoplasmic reticulum. And let me see. A lot of mitochondria. And here you see some smooth ER. Right through here, there's a little sort of like, take a garden hose and cut it up. There's another Golgi. See these here, these little vesicles? They're from the Golgi. And see right here, this thing, anybody know what this is? You're looking down on a Golgi. Whenever you see the picture of Golgi, you always see the cross section, right? What does it look like from the top? It looks like little vesicles that are becoming part of the bigger sort of pancake, and then going into the mature vessels on the other side. So you never really get, this is the pictures you see, especially in intro text, are like the perfect shot that they got. This is right here. This is actually a plastid right there. And here's some more. Here's some rough ER. That's nice rough ER. So you can see the little ribosomes on it. There's some more Golgi right here, looking from the top down or from the bottom up. And you can tell me that these are very metabolically active cells because they've got a lot of mitochondria in it. They've got a lot of Golgi in it. They don't have much of a vacuole, so they're making something. Okay? And they could be making anything from secondary cell wall or primary cell wall material to enzymes. You just can't look at a cell and say, okay, that's from this part of the plant. These are metabolically active cells. And that's about what you can say. Okay, questions? Then let me just finish up just real quickly. To emphasize the fact that we have these different organelles. And what I want to really emphasize is that we can have stuff go from the nucleus, and this is what we're going to be talking about next week, through the ER, into the Golgi, and then all the way out to the cell membrane without ever being basically in the cytoplasm. So it's completely membrane bound from the nucleus till it gets into the cell, membrane. Why? Because if you dump stuff into the cytoplasm, it's going to be ripped apart by enzymes. It's going to be degraded and used. So this transport mechanism is really, really important. Questions? Okay, we're wrapping up early then.